Assalamu alaikum. Well, the second sitting on the smooth muscle is titled as Ionic basis of the action potential. As I have already told you, what is action potential? It is nothing but the ionic transfer across the membrane and which causes the excitable tissues to be triggered very easily. So what happens in the case of a smooth muscle? Yes, you're looking here, depolarization due to entry of the calcium rather than sodium. It's a major difference. In the case of a depolarization of the skeletal muscle, the sodium is important, but in this case, calcium is important. This muscle has more voltage-gated calcium channels than sodium channels. What do you mean by the voltage-gated? That means whenever the electrical potential changes, that causes the opening or closure of those channels, which are known as the voltage-gated. These open and close slowly and responsible for prolonged action. Calcium, along with depolarization of, also causes contraction of the smooth muscle. It's very much important. You're looking here, the explorative and very detailed picture of the smooth muscle having its various contents and the chemicals which are present, and ryanodine receptor is very much important here. This is very much important, present in both cases. Well, these are the beta-1 receptors, the sodium-potassium ATPase, sodium-potassium exchanger, sodium and calcium exchanger. Then the angiotensin. These are the transient outward potassium channels. These are sarcoplasmic reticulum, and these are the kinases and troponin complex, myosin. Tropomyosin is not here. One should be very much clear that tropomyosin is not here. Troponin complex. Electrical activity in multi-unit smooth muscles. Multi-unit smooth muscle, iris and piloerector muscles respond to nerve stimulation which releases acetylcholine and norepinephrine. These do not generate action potential but causes excitatory junctional potential EJP and spread to the entire fiber. Process of excitation, contraction, electromechanical coupling, pharmacomechanical coupling, mechanomechanical coupling. This is the thing. When smooth muscle are excited through depolarization, voltage-gated calcium channels open. Ions enters into the sarcoplasm, stimulate more release of the calcium ions, and calcium ions induced calcium release. You're looking here. Electromechanical coupling, the pharmacomechanical coupling. Pharmacomechanica coupling, smooth muscle are excited by chemical agents mechanism. Chemicals bind to the ligand-gated sodium calcium channels and open it. Influx of the calcium. Ligand comes and there attaches to various receptors. Chemicals bind to membrane receptors, activate second messengers of G proteins through inositol phosphate, causes release of the calcium ions. This second messenger system is very much important. G proteins, it has got a 7-fold and it has got the 3 units, alpha, beta and gamma. Alpha is very much active, which is separated, and it causes the various pharmacologic and various functions on the membrane. Many channels are opened, many kinases they are being activated because of the G protein, which can be a stimulatory as well as an inhibitory. Looking here, membrane depolarization, well, this calcium sarcolemma, then contraction, sarcoplasmic reticulum. See, the calcium in the case of a smooth muscle enters from two sides. One is from the extracellular fluid and other is from the sarcoplasmic reticulum. And when the contraction has taken place, then it has to again come into the sarcoplasmic reticulum. These phospholipase C vasoconstrictor agents, they are very much important. Then comes mechanical coupling. Muscles are excited by stretch, very much important. Stretch sensitive calcium channels are present in the smooth muscles, open and causes influx of the calcium from ECF, extracellular fluid. I have told you that the calcium, in order to cause the gliding and sliding of the actin myosin filaments, that comes from the two sources. One is the ECF, other is from the sarcoplasmic reticulum. Calcium bind with the colmodulin. See, this protein is a difference because here troponin has been replaced by a colmodulin. Colmodulin has got 4 sides for the calcium, and this complex initiates muscle contraction. This colmodulin is very much important in the smooth muscle. Process of muscle contraction. Molecular mechanism is similar to the skeletal muscle, but smooth muscle do not contain tropomyosin and troponin. ये चीज याद रखने वाली है कि स्मूथ मसल में ट्रोपोमाइसिन और ट्रोपोनिन नहीं होती. लाइट चेन ऑफ मायोसिन एक्ट्स एज अ ट्रोपोमाइसिन and called regulatory chain of the myosin. Calcium binding protein colmodulin acts as a troponin. The troponin has been replaced by the colmodulin, which has got 4 sides for the calcium. You are seeing here mechanism of smooth muscle contraction. Cytosolic calcium is increased. Calcium plus colmodulin, activation of myosin kinase, phosphorylation of the myosin light chain, activation of a myosin ATPase, attachment of myosin head with the actin, contraction by sliding filament mechanism. This flow chart is very much important. You must revise it. Mechanism of a smooth muscle contraction: increased cytosolic calcium, calcium plus colmodulin, activation of the myosin kinase, phosphorylation of myosin light chain, activation of the myosin ATPase, attachment of myosin head with the actin, contraction of by sliding filament mechanism. Steps for cross bridges cycling. Calcium combined with colmodulin forms complex, activates enzyme myosin light chain kinase, MLCK, myosin light chain kinase. Phosphorylation of the myosin regulatory chain. The myosin head acquires capability to bind with the actin and forms cross bridge. See, this is that the enzyme which will cause the kinase activation. Steps of the cross bridge: power stroke, formation of actin myosin ADP phosphate complex, conformational change in myosin head, flex towards arm, generate mechanical force power stroke. Actin slide over myosin and causes contraction. Dense bodies are same as the Z lines. Relaxation of the smooth muscle. Calcium pump removes calcium from the ICF to ECF and reverse all stages except phosphorylation of the myosin. You are looking here. These are latch bridges. Dephosphorylated myosin dissociates from actin very slowly, producing slow bridge cycling, maintain tension, tonic contraction. Myosin rapidly dissociates from actin upon binding ATP during each cycle. Relaxed smooth muscle. You are looking here. Dense body, bundle of thick and thin filaments, one relaxed contractile unit extending from side to side to side. And this you are looking here, plasma membrane, thin filament, thick filament, thin filament. This is the thick